Good afternoon. This is Harmony. This is Bio 137, spring semester of 2021. We're in Chapter 10, Nervous System 1. This is lab number 2, Model of a Nerve Impulse. So, all neurons will start, until stimulated, will start at a resting potential, which has an electrical charge of negative 70 millivolts. Each neuron at its resting potential is known as a polarized nerve. This is due to the fact that neurons have a slightly negative internal charge at their resting potential because there is more sodium ions on the outside of the neuron membrane than there are positively charged potassium ions on the inside of the membrane. Even though both ions are positively charged, because there's a surplus of positively charged sodium ions on the outside of the membrane, it causes the internal membrane to be slightly negative charged. And the potassium ions will have a higher rate of outflow than sodium ions will have a rate of inflow, also sustaining this slightly negative internal environment. So when a neuron is stimulated by a presynaptic neuron, a presynaptic neuron will have an axon terminal connecting with the dendrite of the postsynaptic neuron, which is what this is demonstrating. And it will send neurotransmitter signals to create a stimulus to attempt to activate or create an action potential in the postsynaptic neuron. Once the postsynaptic neuron has been stimulated, if it is stimulated sufficiently, it will reach something called the threshold potential, which brings its electrical charge up to negative 50 millivolts. And once this threshold potential has been reached, it creates an action potential. If the presynaptic neuron cannot sufficiently stimulate the postsynaptic neuron, then nothing will happen. But once that threshold potential is reached and an action potential is created, it begins its journey down the length of the axon to reach the axon terminals. It will, immediately upon creation of an action potential, voltage-gated sodium ion channels will open up and allow sodium to flood into the membrane of the neuron at a much higher rate than it was doing so before using the sodium and potassium pumps. This influx of sodium causes something called depolarization, as it brings the inside charge of the neuron up from its original negative, slightly negative charge. It brings it to a positive charge. This is called depolarization. Almost at the same time, these potassium voltage-gated channels open up as well. This is facing the other direction, but it's opening up now. And it allows the potassium to flow out of the neuron at a much higher rate than the pumps will do for them. So this causes the repolarization of the neuron. This is almost instantaneous. Now, upon the opening of the potassium voltage-gated channels, the sodium-gated channels will then close themselves off, reestablishing the rate at which sodium normally enters the neuron, thus repolarizing the cell as well. However, the potassium ion channels don't close at that moment, allowing the potassium to continue flowing outside of the neuron, and this hyperpolarizes the neuron at that point. So now, then the potassium gates will close themselves back up momentarily. This returns the internal resting state of the neuron back to normal. This occurs during a stage called the refractory period, and this disallows the axon from being further stimulated by any type of action potential or impulse. This allows everything to reset itself, allows the axon to return back to its resting potential, while the action potential is conducted along the length of the axon. So this process of the sodium ion gates opening, allowing sodium to flood in, opening the potassium channels, allowing potassium to flood out while simultaneously closing the sodium gates, repolarizing the cell and then hyperpolarizing the cell. This event acts like a chain of dominoes being knocked over in sequence down the length of the axon. So once this section of the axon is finished with this process, it will jump from this node to this node and occur again in sequence down the length of the axon. Now, because this is a myelinated neuron, this has to occur at the nodes of Ranvier. In an unmyelinated one, it would just occur in sequence along the length of the axon. But because it's myelinated, it jumps from node to node until it reaches the axon terminal. Upon reaching the axon terminal, the impulse turns from an electrical impulse to a chemical impulse via the neurotransmitters. The impulse stimulates the neurotransmitter vesicles in the synaptic knobs to open up, thus releasing the neurotransmitter onto the next neuron, and thus passing along the impulse. That should be a sufficient explanation of how a nerve impulse is conducted along the length of a neuron.